Interferon-γ. Interferon-γ IFNG is a dimerized soluble cytokine that is the only member of the type II class of interferons. The existence of this interferon, which early in its history was known as immune interferon, was described by E.F. Wheelock as a product of human leukocytes stimulated with phytohemagglutinin, and by others as a product of antigen-stimulated lymphocytes. It was also shown to be produced in human lymphocytes, or tuberculin-sensitized mouse peritoneal lymphocytes challenged with Mantoux test PPD. The resulting supernatants were shown to inhibit growth of vesicular stomatitis virus. Those reports also contained the basic observation underlying the now widely employed IFN-γ release assay used to test for tuberculosis. In humans, the IFNG protein is encoded by the IFN gene. Function: IFNG, or type II interferon, is a cytokine that is critical for innate and adaptive immunity against viral, some bacterial, and protozoan infections. IFNG is an important activator of macrophages and inducer of major histocompatibility complex class II molecule expression. Aberrant IFNG expression is associated with a number of autoinflammatory and autoimmune diseases. The importance of IFNG in the immune system stems in part from its ability to inhibit viral replication directly, and most importantly from its immunostimulatory and immunomodulatory effects. IFNG is produced predominantly by natural killer cells NK and natural killer T cells NKT as part of the innate immune response, and by CD4 and CD8 cytotoxic T lymphocyte CTL effector T cells once antigen-specific immunity develops as part of the adaptive immune response. IFNG is also produced by non-cytotoxic innate lymphoid cells ILC, a family of immune cells first discovered in the early 2010s. Structure: the IFNG monomer consists of a core of 6 helices and an extended unfolded sequence in the C-terminal region. This is shown in the structural models below. The helices in the core of the structure are numbered 1 to 6. The biologically active dimer is formed by anti-parallel interlocking of the two monomers as shown below. In the cartoon model, one monomer is shown in red, the other in blue. Receptor binding: cellular responses to IFNG are activated through its interaction with a heterodimeric receptor consisting of interferon-γ receptor 1 IFNRON and interferon-γ receptor 2 IFNR2. IFNG binding to the receptor activates the JAK-STAT pathway. Activation of the JAK-STAT pathway induces upregulation of interferon-stimulated genes ISG, including MHC-Roman2. IFNG also binds to the glycosaminoglycan heparan sulfate HS at the cell surface. However, in contrast to many other heparan sulfate binding proteins where binding promotes biological activity, the binding of IFNG to HS inhibits its biological activity. The structural models shown in figures 1-3 for IFNG are all shortened at their C-termini by 17 amino acids. Full-length IFNG is 143 amino acids long. The models are 126 amino acids long. Affinity for heparan sulfate resides solely within the deleted sequence of 17 amino acids. Within this sequence of 17 amino acids lie 2 clusters of basic amino acids termed DUN and D2, respectively. Heparan sulfate interacts with both of these clusters. In the absence of heparan sulfate, the presence of the DUN sequence increases the rate at which IFNG receptor complexes form. Interactions between the DUN cluster of amino acids and the receptor may be the first step in complex formation. By binding to DUN, HS may compete with the receptor and prevent active receptor complexes from forming. The biological significance of heparan sulfate's interaction with IFNG is unclear, however. Binding of the DUN cluster to HS may protect it from proteolytic cleavage. Biological activity: IFNG is secreted by T helper cells, specifically Thon cells, cytotoxic T cells, TC cells, macrophages, mucosal epithelial cells, and NK cells. IFNG is both an important autocrine signal for professional APC in early innate immune response and an important paracrine signal in adaptive immune response. The expression of IFNG is induced by the cytokines IL-12, IL-15, IL-18, and type IIFN. IFNG is the only type Roman II interferon, and it is serologically distinct from type I interferons. It is acid labile, while the type I variants are acid stable. IFNG has antiviral, immunoregulatory, and anti-tumor properties. It alters transcription in up to 30 genes, producing a variety of physiological and cellular responses. Among the effects are: promotes NK cell activity, increases antigen presentation and lysosome activity of macrophages, activates inducible nitric oxide synthase INOS, induces the production of IgG2A and IgG3 from activated plasma B cells, causes normal cells to increase expression of class I MHC molecules as well as class Roman II MHC on antigen-presenting cells to be specific through induction of antigen processing genes, including subunits of the immunoproteasome MEC-lone, LMP2, LM7, as well as TAP and IROP, in addition possibly to the direct upregulation. Promotes adhesion and binding required for leukocyte migration. Induces the expression of intrinsic defense factors, for example, with respect to retroviruses. Relevant genes include TRIM5-alpha, apobic, and tetherin, representing directly antiviral effects. Frames alveolar macrophages against secondary bacterial infections. IFNG is the primary cytokine that defines Thon cells. Thon cells secrete IFNG, which in turn causes more undifferentiated CD4 plus cells, Th0 cells, to differentiate into Thon cells, representing a positive feedback loop while suppressing Th2 cell differentiation. Equivalent defining cytokines for other cells include IL-4 for Th2 cells and IL-17 for Th17 cells. NK cells and CD8 plus cytotoxic T cells also produce IFNG. IFNG suppresses osteoclast formation by rapidly degrading the rank adapter protein TRAF6 in the rank-rankle signaling pathway, which otherwise stimulates the production of NFKB. Activity in granuloma formation. A granuloma is the body's way of dealing with a substance it cannot remove or sterilize. Infectious causes of granulomas. Infections are typically the most common cause of granulomas include tuberculosis, leprosy, histoplasmosis, cryptococcosis, coccidioidomycosis, blastomycosis, and toxoplasmosis. Examples of non-infectious granulomatous diseases are sarcoidosis, Crohn's disease, borreliosis, giant cell arteritis, granulomatosis with polyangiitis, eosinophilic granulomatosis with polyangiitis, pulmonary rheumatoid nodules, and aspiration of food and other particulate material into the lung. The infectious pathophysiology of granulomas is discussed primarily here. The key association between IFNG and granulomas is that IFNG activates macrophages so that they become more powerful in killing intracellular organisms. Activation of macrophages by IFNG from Thon helper cells in mycobacterial infections allows the macrophages to overcome the inhibition of phagolysosome maturation caused by mycobacteria to stay alive inside them. The first steps in IFNG-induced granuloma formation are activation of Thon helper cells by macrophages releasing IL-1 and IL-12 in the presence of intracellular pathogens and presentation of antigens from those pathogens. Next, the Thon helper cells aggregate around the macrophages and release IFNG, which activates the macrophages. Further activation of macrophages causes a cycle of further killing of intracellular bacteria and further presentation of antigens to Thon helper cells with further release of IFNG. Finally, macrophages surround the Thon helper cells and become fibroblast-like cells walling off the infection. Activity during pregnancy. Uterine natural killer cells secrete high levels of chemoattractants such as IFNG in mice. IFNG dilates and thins the walls of maternal spiral arteries to enhance blood flow to the implantation site. This remodeling aids in the development of the placenta as it invades the uterus in its quest for nutrients. IFNG knockout mice fail to initiate normal pregnancy-induced modification of decidual arteries. These models display abnormally low amounts of cells or necrosis of decidua. In humans, elevated levels of IFNG have been associated with increased risk of miscarriage. Correlation studies have observed high IFNG levels in women with a history of spontaneous miscarriage when compared to women with no history of spontaneous miscarriage. Additionally, low IFNG levels are associated with women who successfully carry to term. It is possible that IFNG is cytotoxic to trophoblasts, which leads to miscarriage. However, causal research on the relationship between IFNG and miscarriage has not been performed due to ethical constraints. Production. Recombinant human IFNG, as an expensive biopharmaceutical, has been expressed in different expression systems including prokaryotic, protozoan fungal yeasts, plant, insect, and mammalian cells. Human IFNG is commonly expressed in Escherichia coli, marketed as Actimuner. However, the resulting product of the prokaryotic expression system is not glycosylated with a short half-life in the bloodstream after injection. The purification process from bacterial expression system is also very costly. Other expression systems like Pichia pastoris did not show satisfactory results in terms of yields. Therapeutic use. Interferon-γ is approved by the U.S. Food and Drug Administration to treat chronic granulomatous disease (CGD) and osteoporosis. The mechanism by which IFN-γ benefits CGD is via enhancing the efficacy of neutrophils against catalase-positive bacteria by correcting patients' oxidative metabolism. It was not approved to treat idiopathic pulmonary fibrosis (IPF). In 2002, the manufacturer Intermune issued a press release saying that phase Roman 3 data demonstrated survival benefit in IPF and reduced mortality by 70% in patients with mild to moderate disease. The U.S. Department of Justice charged that the release contained false and misleading statements. Intermune's chief executive, Scott Harkonnen, was accused of manipulating the trial data, was convicted in 2009 of wire fraud, and was sentenced to fines and community service. Harkonnen appealed his conviction to the U.S. Court of Appeals for the Ninth Circuit and lost. Harkonnen was granted a full pardon on January 20, 2021. Preliminary research on the role of IFNG in treating Friedreich's ataxia conducted by Children's Hospital of Philadelphia has found no beneficial effects in short-term 6 months treatment. However, researchers in Turkey have discovered significant improvements in patients' gait and stance after 6 months of treatment. Although not officially approved, Interferon-γ has also been shown to be effective in treating patients with moderate to severe atopic dermatitis. Specifically, recombinant IFNG therapy has shown promise in patients with lowered IFNG expression, such as those with predisposition to herpes simplex virus and pediatric patients. Potential use in immunotherapy. IFNG increases an anti-proliferative state in cancer cells while upregulating MHC-I and MHC Roman II expression, which increases immunorecognition and removal of pathogenic cells. IFNG also reduces metastases in tumors by upregulating fibronectin, which negatively impacts tumor architecture. IFNG is not approved yet for the treatment in any cancer immunotherapy. However, improved survival was observed when IFNG was administered to patients with bladder carcinoma and melanoma cancers. The most promising result was achieved in patients with stage 2 and 3 of ovarian carcinoma. On the contrary, it was stressed interferon-γ secreted by CD8-positive lymphocytes upregulates PD-L1 on ovarian cancer cells and promotes tumor growth. The in vitro study of IFNG in cancer cells is more extensive, and results indicate anti-proliferative activity of IFNG leading to the growth inhibition or cell death. In addition, It has been reported that mammalian glycosylation of recombinant human IFNG, expressed in HK293, improves its therapeutic efficacy compared to the unglycosylated form that is expressed in E. coli. Interactions. Interferon-G has been shown to interact with interferon-gamma receptor 1 and interferon-gamma receptor 2. Diseases. Interferon-G has been shown to be a crucial player in the immune response against some intracellular pathogens, including that of Chagas disease. It has also been identified as having a role in seborrheic dermatitis. IFNG has a significant antiviral effect in herpes simplex virus IHSV infection. IFNG compromises the microtubules that HSV relies upon for transport into an infected cell's nucleus, inhibiting the ability of HSV to replicate. Studies in mice on acyclovir-resistant herpes have shown that IFNG treatment can significantly reduce herpes viral load. The mechanism by which IFNG inhibits herpes reproduction is independent of T cells, which means that IFNG may be an effective treatment in individuals with low T cells. Chlamydia infection is impacted by IFNG in host cells. In human epithelial cells, IFNG upregulates expression of indolamine 23 dioxygenase, which in turn depletes tryptophan in hosts and impedes chlamydia's reproduction. Additionally, in rodent epithelial cells, IFNG upregulates a GTPase that inhibits chlamydial proliferation. In both the human and rodent systems, chlamydia has evolved mechanisms to circumvent the negative effects of host cell behavior. Regulation. There is evidence that interferon gamma expression is regulated by a pseudo-knotted element in its 5-UTR. There is also evidence that interferon gamma is regulated either directly or indirectly by the microRNA myR-29. Furthermore, there is evidence that interferon gamma expression is regulated via gaped-in T cells. This interaction takes place in the 3' where binding of gapt prevents the translation of the mRNA sequence.